Have you ever stopped to think about one of nature's greatest magic tricks? I mean, how a single, tiny, almost invisible cell can somehow transform into a living, breathing, hopping frog? It sounds impossible, right? Well, let's dive into this absolutely incredible story.
Welcome to Silverlight Education. In this lecture, we're going to be covering BZYCT 133 course, specifically Block 4, Unit 14.
Okay, so let's get right back to that central mystery. This is a question that has captivated scientists for literally centuries. How on earth does one single, ordinary-looking cell become a frog? It's one of nature's true marvels, and today, we are going to break down every single step of this astonishing journey. And here's the part that'll blow your mind. This whole incredible process actually begins long before the first cell even divides. It all starts with a complete blueprint, a pre-existing plan that's already tucked away right inside the egg. So, what is this blueprint? Well, it's all about something called polarity. You can think of the unfertilized egg as having a built-in map. The top half, the pigmented animal hemisphere, is totally distinct from the bottom half, which is the yolk-filled vegetal hemisphere. This simple top-to-bottom layout is the very first, most fundamental step in designing the entire body. And then comes this moment of incredible drama. A single sperm enters the egg, and it triggers a stunning event. The egg's outer layer, its cortex, rotates precisely 30 degrees. And in that one single movement, the entire body plan is locked in. Just like that, it establishes where the frog's back will be and where its belly will be. One tiny moment, and the body's entire map is set. Amazing. Okay, so the blueprint is ready. The major directions, top, bottom, back, front, are all defined. But how does the embryo get the billions of cells it needs to actually build something? Well, let's move on to the construction phase and see how it multiplies its workforce. The answer is a process called cleavage. Now, this is not your normal, everyday cell division. This is division on hyperdrive. It's a series of incredibly rapid splits that happen without any of the cells actually growing bigger. The embryo just keeps dividing its existing material, turning one large cell into a hollow ball made of thousands and thousands of smaller cells, which we call blastomeres. And those first three divisions, they are an absolute masterpiece of geometric precision. The very first cut is vertical, and it separates the future left and right sides. The second cut is also vertical, creating 4 perfect quadrants. And then the third cut is horizontal, neatly separating the smaller cells at the top from the larger yolk-filled cells at the bottom. You see? This isn't random at all. It's a highly organized construction plan. Now, after about 12 of these divisions, something truly pivotal happens. It's called the mid-blastula transition. You see, up until this point, the embryo has just been running on autopilot, using instructions and materials left behind by the mother. But now, at the MBT, the embryo's own genes switch on. This is a critical handover. It's the moment the embryo becomes truly self-reliant, taking command of its own development for the very first time. With a full workforce of cells now ready to go, we enter what is arguably the most dynamic and critical stage of development, gastrulation. Think of this as a great migration, an epic journey where thousands of cells move to find their ultimate purpose in the body. So what is gastrulation? It's a massive, highly organized movement of cells from the embryo's surface to its interior. This whole dance begins when a special group of bottle cells on the surface change their shape. They constrict at their tops and sink inward. This creates a little opening, a crescent-shaped slit called the blastopore, which acts as the main gateway to the inside. As all these cells start pouring inward, they perform an incredible maneuver called convergent extension. And to get your head around it, just imagine this. Thousands of cars spread out across many, many lanes of a highway, all needing to merge perfectly into a single flowing lane. That is exactly what these cells are doing. The tissue narrows and lengthens dramatically, all through this highly coordinated movement. And the result of this great migration? The formation of the 3 foundational layers of the body. The outer ectoderm, that's going to become the skin and the entire nervous system. The middle mesoderm will form all the muscle, bone, and organs like the heart. And the inner endoderm, that creates the lining of the gut. Every single part of the adult frog originates from one of these 3 layers. So, with the body's foundational layers now in place, the next major task is to build the command center, the central nervous system. This happens through a process called neurulation, and honestly, it's a stunning feat of what you could call biological origami.
Yeah, you can really think of it like folding paper. It starts with a flat sheet of ectoderm on the back of the embryo, which thickens into what we call the neural plate. Then, the edges of this plate rise up, creating neural folds. These folds then bend toward each other, they meet in the middle, and fuse together, forming a perfect tube that then separates from the surface to become the future brain and spinal cord.
But wait, how do the neural tube and the future skin separate so cleanly? It seems like they should just be stuck together, right? Well, the secret is in their molecular glue. The surface cells, the ones that become skin, use a molecule called E-cadherin to stick to each other. But the neural tube cells, they switch to a different glue, N-cadherin. And because these two glues don't mix, the tissues separate perfectly. It's just an incredibly elegant solution.
And now we finally arrive at the final and by far the most dramatic chapter of our story, metamorphosis. This isn't just a little change. It is a complete and total reconstruction, where an aquatic tadpole re-engineers itself from the ground up to become a terrestrial frog. And the scale of this change is just staggering. I mean, think about it. A tadpole breathes with gills. A frog uses lungs. A tadpole swims with a tail fin. A frog hops on powerful legs. A tadpole is a plant eater. A frog is a carnivore. Even its waste products change from ammonia to urea. It is genuinely like two completely different animals are living inside one single lifetime.
So what kicks off this radical overhaul? A very precise hormonal chain of command. It all starts in the brain with the hypothalamus releasing a hormone. This tells the pituitary gland to release another hormone, which in turn signals the thyroid gland. The thyroid then floods the body with thyroid hormones. And that is the master signal that gets this incredible transformation started.
And through all of this, division, migration, folding, and complete transformation, we are reminded that this entire complex, unbelievable journey began with just one single cell. You know, this journey from a single cell is so much more than just biology. It's a living testament to how profound, almost unbelievable complexity can arise from the very simplest of beginnings. And it really makes you wonder, doesn't it? What other hidden blueprints are out there, quietly shaping the world all around us?
We will meet you in the next lecture.